Hello, my name is Sanat Myti. I am a second year graduate student. I'm working with Dr. Byung Renso. She is a specialist in the RNA biogenesis area. So my research topic is discovering the role of diadenosine tetraphosphate in the formation of RNA protein complexes in innate immunity. We know that RNA um, gene expression is the way by which innate immunity combat the infectious diseases by some antimicrobial pathway transcriptions and adaptive immunity. So for all of those steps, transcription is the first and foremost step that is well scrutinized and well studied. But there are some other steps that requires for the gene expression for innate immunity. It's called post-transcriptional modification of the pre-mRNA. That is not that much well studied. So I am working on that special area because each in special specific gene expression actually for it's for the specific infectious microorganism that needs specific gene expression. So we know that diadenosine tetraphosphate is an alarm1 which elevates by the phosphorylation of lysyl tRNA synthetase from multisynthetase complex. Upon phosphorylation of lysyl tRNA synthetase, it's translocate into the nucleus and then form the AP4A, which is called diadenosine tetraphosphate. Upon elevation of diadenosine tetraphosphate, it regulates the gene expression by up and down regulation of innate immunity specific genes. We have seen that it activates the MITF mediated transcription. In 2021, one group published a paper in Nature Communication, and they have shown us that AP4A interact with some pre-mRNA processing machineries. But they did it in the cell lysis, which has a great chance to interact spontaneously with lots of proteins. That's why we wanna avoid those things. That AP4, we need to know which specific protein interact with AP4A, and that help to regulate the gene expression in innate immunity. That's why we went for checking the stability of AP4A in the cellular condition upon the AP4A hydrolase knockdown. And then we checked that AP4A is stable up to 1 hour in the chronic myelogenous leukemia cells. And then we go for the mass spectrometric analysis of the AP4A interactome. We found that AP4A associates with some H and RNPs, which is the splicing factor for the pre-mRNA. For that reason, we go for checking naturally producing AP4A interactome in the chronic myelogenous leukemia cell. Again, then we treat the cell with dinitrophenol and then check the H and RNPs complex. Then we have seen that H and RNPs complex dissociates with some H and RNP S. That means most probably AP4A has some common structural motif with H and RNP's complexes. That's why we'll go for checking the splicing events for the specific H and AP4A interactome with H and RNPs, and we will check the binding efficiency with H and RNPs and how they affect the gene expression for the innate immunity. We also check for the skipping event for CD45 in the upon T cell activation because it's mediated by the H and RNPL. So my future direction will be on those parts.